All right, good afternoon, everybody. I think we can start. It's 1 minute past 12. Please, I just want to make one announcement. It came to my attention that based on the instructions from the Vice Dean, you're supposed to register attendance downstairs as well. All right? I think it's because it's a practical session, so that makes sense. So you need to go, once you go to the lab, make sure you do the face scan or facial recognition. All right, so punch in basically your attendance. All right, yes? That's all right, we understand. We wanted you to either take attendance here or downstairs. We thought that since you're all coming here first, you could do it here, but recently Dr. Yasin, the Vice Dean, said no, it should be downstairs. So the instructors downstairs are aware, and they will give you the opportunity to basically punch in, all right, once you go downstairs. If you can certainly do it here as well, but I believe that in the registration, the session is registered under the lab. It's not registered in this lecture hall, so probably, yeah, that could be the reason behind it. But no worries, things are good, don't worry, these things happen. All right, so today's session, we're going to talk about some basic techniques in biochemistry, lab techniques in biochemistry, and these techniques are typically involved in separating things from another. So whether it's chromatography or centrifugation, you'll see that there is a common theme, and that they involve separating molecules, separating chemicals maybe, or this could be separation of proteins or cells. We will also talk about a very important, let's say, technique in biochemistry, and that is spectrophotometry. So I'd like to briefly begin by going over the lab objectives, and I encourage you to do the same when you go downstairs, because if you read the objectives, you'll know what to focus on, what you need to learn about, and what outcomes should you come out with at the end of the session. So you should be able to identify certain terminologies, new terminologies, and that is chromatography, spectrophotometry, centrifugations. Those are the three main things that I'll be talking about in this session. These are the three main things that you're going to be seeing when you go downstairs in the lab. There's a fourth thing that is not listed here, which is you are going to practice as well taking a urine dipstick, just the principle behind urine dipstick. So that is a stick that we dip in the urine. That is why it's called dipstick. And there's color change, and that color change can tell you something. Usually can tell you if there is sugar in the urine or there's excess protein, etc. But so these are the four main things that we will be briefly describing, but the three at the top are the ones that you need to focus on. All right, so you need to learn certain terminologies related, for example, to chromatography. These new terminologies include stationary phase. What do we mean by stationary phase? Mobile phase. What do we mean by that? Affinity of component to solvent. Adsorption, all these things, adsorption, affinity, mobile phase, stationary phase, these are terminologies used in chromatography, okay? So I'm going to spend more time on chromatography, and in that you will learn these terms: stationary phase, mobile phase, affinity, and adsorption. When you learn about chromatography, you're also supposed to learn how to perform it, that's downstairs, and how to calculate something known as RF, or retardation factor. Downstairs, you will also be in the lab, you'll also be performing urine dipstick. You'll be provided with a kit, asked to do it, and then read or interpret the results. You will separate mixtures provided to you by centrifugation. So again, once again, I am going to go through these processes: chromatography, spectrophotometry, centrifugation. What do they mean? But you will go downstairs and you'll practice these techniques as well, and measure the provided solution by spectrophotometer. So this is what you're going to do downstairs, all right? I'll just talk about the concepts and just the theoretical part. All right, so let's begin. So what do you see here? Can you tell me, has anyone ever seen this, something like this happening? Yeah, it happens, right? You're writing something, you get a drop of some kind of solution. This could be water, maybe alcohol, maybe something else, a drop of oil. But regardless, sometimes you may see something like this. So this is basically a principle that we refer to as chromatography, or paper chromatography in this case. So essentially the blue dye is the original dye, and in the presence of the paper, in the presence of that liquid, that solvent, somehow that dye was separated into its original mixture or components, let's say. So that tells you that probably the blue ink is really a mixture of other dyes. Maybe it's a mixture of green and pink combined together. So in a very basic way, this is what chromatography means. It is a separation technique. And chroma is a word it's a Greek word that means color, whereas graphy means writing. So when you combine them, that's what it is. But essentially, it is a separation technique whereby you can separate molecules, or you can separate different components. And if you want to separate things, you can imagine that this separation technique, it means you understand the real composition of a given solution. You can understand that this can have a lot of applications. Maybe in the food industry, I want to see what is the composition of this juice or this any other beverage or something. Could be used in the industry, not just in food, but in the industry like the paints and dyes. But it can also be used in medicine. We can separate we can take a urine sample and then separate the different components, see what is there in the urine, and so on. So you can understand that this could be applied to as well body fluids, hence you can use this separation technique for diagnostic purposes. So for separation to occur in chromatography, you need two things. You need a mobile phase and a stationary phase. In the picture that I showed you, the mobile phase is the water, the drop of water, okay? It is basically the solvent that will carry your molecule, all right? And the stationary phase is what? Can you guess? Is the paper itself. And the paper basically is what the molecules will go through or will pass through. So it passes through the paper, and what carries it as it's passing through the paper, what carries it is the solvent, it's the water. So the stationary phase, by definition, is what is the phase which the molecules pass through, whereas the mobile phase is what the molecules are carried by. And for separation of components to occur, this depends on several factors, but there are essentially two main things. The affinity to the solvent. The solvent is also known as the mobile phase or the mobile phase. And number two, adsorption to stationary phase. How much, let's say, adsorption, as you can say, how much friction. You can think of it this way. So affinity to the solvent is how much the molecule likes the solvent. If there is great affinity, it likes it a lot, it can easily be carried by it. So this, you can imagine that separation will be better. And vice versa. If the affinity is not that great, the separation, you can understand, will be affected negatively. Same thing with adsorption. If there's a lot of adsorption on the paper, نوع من الاحتكاك, you can think of it, then the amount or the distance that it'll travel, the separation will be more difficult because it's kind of more attached to it. So those two things will affect the separation of components, the separation of molecules in chromatography. All right, so do we know what the stationary phase is? So by definition, you need to know, please, stationary phase, what is meant by it? Mobile phase, what do we mean by it? And those two important determinants of separation, which is affinity to solvent and adsorption to the stationary phase. So affinity to the mobile phase and adsorption to the stationary phase. There are different types of chromatography. You do not need to know the different types, just that these different types do exist. There's column chromatography, ion exchange, affinity, thin layer chromatography, gas chromatography, and the list goes on. And the one that I'm going to talk about today is the simple one, and that is the paper chromatography. When you go downstairs, you're going to conduct paper chromatography, all right? You're going to do that exercise. Please ask yourself, in this experiment, what is the mobile phase? What is the stationary phase? What are those two factors that determine how much separation will happen, if it happens? So please, these concepts are very important to bear in mind. So this is an example of what you will be doing. You can take a filter paper, because you're going to be doing paper chromatography. You're going to put a start line. You can put a line here and then a dot. You'll have an ink provided. Then you'll dip this in a beaker with some water. So the water will act as what kind of phase? Is it stationary phase or mobile phase? It's mobile phase. And what is the stationary phase in this case? It is the paper. So you'll wait and you'll basically see, first of all, you'll see that the water will get absorbed by the filter paper and it'll reach a maximum. This could be here. So here it's a lighter shade. Here it's a darker shade. So the water has soaked this area of the filter paper. If this is the maximum, you can say that this is the solvent front, or this is the distance traveled. So what is the distance traveled? You soaked it here and it traveled all the way over here. All right? And then you should see some separation. This is an experiment where they put an ink spot here, and that ink spot, because it is not pure ink, it is not pure black, let's say, it's actually a mixture of other colors, then you will see the separation. You'll see red, blue, and yellow. So then you can assume that there's probably 3 different components in that drop of ink that was used. So why do I want you to do a start line and a finish line or whatever? Because, once again, this is just a much better visual. If you have your beaker, you'll somehow attach this filter paper, and you'll put a dot. You'll draw a line, which will be your start line, and try not to dip the filter paper too much. So make sure that the water is below the start line, and this is your drop of ink. Eventually, the water will get absorbed by the filter paper, and this is the solvent front, basically, or the water front in this case. All right? As the solvent travels up or flows upwards, you will see separation of different components within that initial drop. So if this is the sample at origin, once you dip it here, perform the paper chromatography, you should see it being separated to different components and different colors. All right? So it just takes time. So now, do we know what is mobile phase, what is stationary phase? Can you tell me if this red dot traveled all the way here, and then the blue dot only traveled here? What can you understand? Which one do you think has higher affinity to water or to the solvent? The red one, excellent. Which one do you think has more adsorption to the paper? The blue one, exactly. So this is what we mean, that the affinity to solvent, adsorption to the paper will determine how much separation will occur, all right? And in this case, how far these components will travel, all right? So this is basically, again, another image. You'll do something like this. You'll attach a filter paper. You'll insert it into water, and you will watch your specimen or your initial mix. You'll watch it separate into its original composition. So you'll have mix A could give you blue and purple. Mix B can give you green and blue and purple, and so on. So please go downstairs, do this paper chromatography, and understand that it is a separation technique, and understand that for the separation to occur, you should have a stationary phase, you should have a mobile phase. And stationary phase is what the molecules pass through. The mobile phase is what carries these components, all right? And we said the two determinants of separation, affinity and adsorption. Why did I say affinity and adsorption and how much distance the solvent traveled and measure this and measure that? It's because we want you to calculate something known as retardation factor. So let's say this is your original sample and it traveled, let's say, all the way here. And the solvent you used will travel all the way here. So this would be your starting line. This would be the finish line. The finish line is the maximum where the water can reach, or the solvent, all right? It could be any solvent. So it doesn't always have to be water. It could be alcohol. It could be something else. So what you will do when you go downstairs, you will measure the total distance traveled by the solvent, and you will measure the total distance traveled by the component. And you will divide the distance traveled by component over the total distance traveled by the solvent. And that will give you the retardation factor for that component, all right? And so you can imagine if the retardation factor is great, what does that mean in terms of solubility and affinity to the solvent? It's high. If it has high affinity to the solvent, or in other words, it has high solubility, then it will travel more, so that value becomes larger, and therefore the RF will be larger. Whereas if this travels less, then the retardation factor will be smaller, right? If you have a dot that is here, for example, then you can imagine that A is smaller and the RF value will be smaller. And if the RF value is smaller, you can understand and you can interpret this result as what? High adsorption, and what about solubility? Small, all right? So if RF is great, you can imagine that solubility is probably great, or the adsorption is very small, or both. Whereas if the RF factor is small, it means it can't travel as much, it can't separate as far. This could be because it's not very soluble in the solvent, or it doesn't have very great affinity to it, or the adsorption to the paper, or the adsorption to the stationary phase is high, all right? So please learn how to calculate retardation factor and how to interpret it in terms of affinity to the solvent or solubility, and in terms of adsorption. You will also do another type of chromatography, but I don't know how much time, so it all depends on how fast we can get you downstairs. So basically, you'll have a sample mixture, and you'll add to it something called an elution buffer. This will help with the separation, and you should see this mixture separating to its different components, but in this case we're not using really water, we're using a column. We're not using paper, we're using a column chromatography technique. We have a video here, you can have a look, but all right, there you go. So it'll look something like this, but as you can see, the dripping of the solution is taking a long time, and therefore it might be time consuming. I don't know how much time you'll be able to spend downstairs, but you should be able to separate the given mixture. They'll give you a mixture downstairs, and you should see the different colors and different compositions, and you can collect them in different tubes. But that typically takes it takes time. It might take an hour. I honestly don't know precisely how long it would take, but so you can have these different components separated from your original mixture. All right, but this is a different chromatography technique. It is called the column chromatography. All right, so I spent a fair good amount of time explaining about chromatography. We learned the two main components of chromatography, the stationary phase, the mobile phase. We talked about the two important factors that determines how much separation will occur. We learned about a new terminology known as retardation factor. What is retardation factor? What does it mean if it's small? What does it mean if it's big? Now I'd like you to forget a little bit about chromatography and focus on something called, it's kind of another separation technique, but it's different. It's called centrifugation. So for chromatography, I will separate mixtures into their original compositions. Here, we're using the density. We can separate things based on their shape and their mass and their density, and we can separate them. And the best example would be having a blood sample. So if you have a blood sample, for example, you can use centrifugation. You can put the blood in a tube. That tube, you will put it in a machine called the centrifuge machine. It'll spin it so fast. Imagine I have a rock at the end of a string, and I'm rotating this way. So the same thing, that's called centrifuge or centrifugal force. All right? So that force, centrifugal force, when you do it fast enough, the particles that have higher density will settle at the bottom. The things that have lower density will be at the top. All right? And we do this for blood. I'm just going to show you the machine, how it looks like. So imagine you take a blood tube, you put it in the centrifuge machine. It looks something like this. You will do this downstairs. You will see a centrifuge machine. Just have a look. So you put a tube. If you have only one tube, opposite to it, so if you put a tube here, opposite to it exactly, you have to put another tube, kind of the same weight or the same mass, because it has to be balanced. If it's not balanced, as this spins so fast, it could break. But if it's balanced, it could spin so fast with no problem. But if you only put one tube, that's dangerous. Or if it's uneven, if you put two tubes side by side, this means in your disc thingy, one side will be heavy and one side will be empty. So this will make the spinning tilted in a tilted manner. And that spinning in a tilted manner can break the centrifuge machine. So we typically put samples opposite to each other so that it's always balanced. And if you only have one blood specimen from a patient, what you can do is on the opposite side to your blood specimen, just put a tube with water in it, for example. All right, and you should make them approximately the same weight or the same size and so on. So you put it, it spins and spins, and that's basically will give you something that looks like this. So you can, if you're doing it with blood, you'll have the upper layer with plasma, and the bottom part is more dense. It's your red blood cells, and in the middle there's something called buffy coat, and that contains the white blood cells and the platelets. And the combination of buffy coat and the erythrocyte is also known as hematocrit, and it's approximately 45% of the total volume of blood. All right, so this is blood before centrifugation. This is blood after centrifugation. The top part, what do we call it? Plasma. The middle part, the buffy coat, I know it's not clear in here, but sometimes if you want to do PCR on DNA, right? So you'll get the DNA from white blood cells. You can't get it from red blood cells. Can you? Because red blood cells don't have nuclei. Same thing with platelets. But what we do is we aspirate the fine white cloud-like layer in the middle. You might not see it clearly here, but you can insert a tip and gently suction it out, and then you can lyse it and break it and process it for DNA extraction and PCR. All right, so this is how it would look like. I do want to tell you though, usually you will get this nice separation if you collect blood in a special tube that is treated with what? Anticoagulant. Because when you collect blood from a patient, if your tube does not have an anticoagulation or chemicals for anticoagulation, so it doesn't have anticoagulant, it will clot. It's something spontaneous. When there is stasis, the blood is not moving, this will activate the coagulation cascade and you'll get a blood clot. Okay, بس نبي جلة في الدم. It will show this nice separation if you collect the blood in a tube that contains anticoagulants that prevent coagulation. All right? So what do you think is in the plasma, which is the top part? There's water, proteins, nutrients, electrolytes. In the buffy coat, it's white blood cells and platelets, and the bottommost part, it's the erythrocytes or the red blood cells. All right. So this is again centrifuge. And remember, so centrifuge is used as a separation technique, and the best example is blood. But I can use it if I'm growing cells. I could be growing cells in a dish, and you know how we have to add a media to the dish, right? So if I want to wash the cells or collect them and plate them somewhere else, I will harvest the cells, and they will be floating in a solution, in a liquid. I want to get rid of this liquid so I can spin it. And the bottom part will give me the pellet, which contains my cells only, and the supernatant, the top part, which only contains the media. So I can simply take it and pour it out, and I'll end up with the cells only. So this is a separation of cells, for example. So it's not only separation for the blood. So this is what we use centrifuges for, all right? And so this is basically what would happen if you collect blood in a regular tube that is not treated with anticoagulants. So it will typically, after you set it for some time, you put it on your table, on your bench space in the lab, it'll clot. You'll have a blood clot here, and the rest will be serum. However, if it is treated with anticoagulants, then it will not clot spontaneously, and when you spin it, you'll have the nice separation where you'll get plasma, buffy coat, and red blood cells. Now the next question is, what is the difference between serum and plasma? So, yes. It's plasma. Serum is plasma without the fibrinogen, without the clotting factors, because here a blood clot happened. And for a blood clot to happen, usually it activates a lot of proteins and clotting factors, including fibrinogen. So then you will consume them, you will use them in this clot. So this liquid part is exactly the same as plasma, but without the proteins and clot that are involved in coagulation. So without the clotting factors, and more importantly, without the fibrinogen, which is a component of this blood clot. All right? So if you have a tube, you collect blood in it, and if you assume that the tube has no anticoagulant, what will happen to the blood? Blood eventually? It will clot. And when there's separation, and eventually it will separate, even if you take plasma and you put it without the centrifuge, but it's just going to take a longer time, you will get separation. Put it in the fridge. We do it for research purposes. Sometimes we collect bloods. Sometimes we spin it right away. We spin it as in we centrifuge it, but sometimes we just put it in the fridge, and it will naturally separate into plasma and the red blood cell composition. But this will show much better separation. And of course, it's much quicker. You only need to spin for like 3 minutes, maximum 5 minutes. All right? So if you put it in a tube without any anticoagulant, then what you'll end up with is with a blood clot, and the top part will be serum. Okay, so please differentiate between serum versus plasma. All right. I talked about chromatography. I talked about centrifugation as another separation technique. Now I'd like to talk about, it's not really a separation technique, but because it's something called spectrophotometry, and we use it to measure usually concentrations of things. And a very good example would be concentration of protein, for example. This could be protein in a specimen that you have. It could be protein in a urine sample. It could be protein in your blood. It could be hemoglobin in your blood. And the principle behind spectrophotometry, it depends on basically measuring, it depends upon measuring the amount of light that is absorbed and or transmitted in a specific wavelength. What does that mean? Basically, imagine this is your container that has a solution. It could be a solution that contains some proteins, and you want to see how much protein is in this sample. So I'll put it in this device known as spectrophotometer. I'll put my specimen inside. And inside the spectrophotometer is a light source. This white light will shine through a prism, and it will dissociate the light into different wavelengths. All right? So you'll have, different beams of light with different wavelengths, and then you can select which wavelength would you like to shine on your solution. And this will depend on your specimen. So probably there will be a particular wavelength that could be absorbed by the particles, the proteins that are inside your specimen. So you need to select. You don't want to shine the whole light because this will scatter light. So you select a certain wavelength that will be absorbed by the proteins inside your solution. And on the other side, there's a detector that detects the transmitted light. So think of it this way. This is how much you're shining, and this is how much is coming out, which means it's less, it's faded. So the amount that is being detected by the detector or the photometer will be less. So this depends on how much particles are in the solution, how much particles or how many particles are absorbing the light at that particular wavelength. So if you have a lot of particles and they're all absorbing a lot of those wavelengths, what do you think? The detector will detect a lot of light or little light? Little light. So it's kind of like a calculation and an estimation, knowing how much light you're transmitting. Sorry, how much light is the incident light, they call it. It's being, you shine it on your specimen. From that, some of it will be absorbed, some of it will be transmitted, and the photometer will give you a reading. All right? So this gives you an idea about basically concentration of your solution using this technique. All right, so what do we call this? Spectrophotometer. So it measures light absorption or transmission in a specific wavelength using a photometer. So the photometer is what will measure the light transmitted and the light intensity. And usually the reading here, it's usually expressed as absorbance. So if there's a lot of particles or if there's high protein concentration here, there will be higher absorbance of light and less transmission of light. All right, this is essentially the same thing. So you have your light source, then you have your prism, and then different wavelengths, and then you, through a slit, you have a wavelength selector. You'll select the appropriate wavelength for your specimen, and this is the incident light, this is the transmitted, and this is the photometer that will read the absorbance. All right, so you have different, they come in different sizes, different shapes. Please go downstairs and have a look. Now, before I release you, I'd like to talk about a dipstick. That is another exercise that you'll do. Chromatography, we separate components from each other for mixtures, and we're identifying what is this mixture composed of. We talked about centrifugation, also as a separation technique. We talked about spectrophotometry to measure proteins, maybe concentrations of proteins and densities. The last thing I want to talk about is urine dipstick, and it can basically detect. You have this stick with membranes, small squares. Each membrane is different. It has different chemical composition, and every one of them can react with something very specific. One of them may react with ketones in the urine. One of them will react with sugar or glucose in the urine, and so on. Once they react with these chemicals in the urine, they will change color. And then you will see this color change, and you will interpret it based on your reference. So there will be a reference. It will tell you if there's a lot of glucose or no glucose based on the color change. So imagine, so this is your urine dipstick, and each one of them is a different, each of those squares is a different, basically, membrane with different properties that will detect something different. So this could be pH. It will tell you if the urine is acidic or alkaline, and you will be able to know by the change in color. What color did it change into once you dipped it into the urine? So please go downstairs and enjoy the lab, and don't forget to register attendance downstairs in the lab, please. Thank you.